Welcome to Data and Biotech, a podcast from Cornell where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Welcome back to Data and Biotech. I'm Ross Katz, Principal and Data Science Lead at Corden. Here's a question biotechs encounter more and more frequently. You've got years of assay data scattered across hard drives, unlabeled folders, and random directories that only one person knows about. Is that data worth anything? 2 years ago, the answer was probably not. The cost of organizing it was high, and there was no obvious payoff. But biofoundation models are starting to change that in a fundamental way. These models can take heterogeneous data across different assays, different modalities, different experimental contexts, and make use of it, which means all that stuff you might have treated as disposable might turn out to be one of your most valuable assets. Jesse Johnson is the founder of Merelogic, a software consulting firm that works directly with biotech labs on exactly this problem. He's not building foundation models himself. He's on the other side of the coin, helping the companies that need to use them figure out how to get their data house in order without over-engineering it. That combination of technical depth and client-facing pragmatism gives him a grounded perspective on what works when the rubber meets the road. Jesse, welcome back to the show.
Yeah, thanks for having me. I'm glad to be back.
Awesome. Well, Jesse, for listeners who didn't hear you when you were last on Data and Biotech, as you told us then, you went from doing data quality engineering at Google, where the data is clean and the systems are deterministic, into biotech, where the data is messy and the systems are, biological. So how did that transition change your fundamental assumptions about what good data infrastructure looks like?
Yeah, well, I think there's some major differences in the way data is collected and managed in biology that it definitely took me longer than it probably should have to realize. But I think the main difference from an organizational perspective is that in the tech world, the engineers have complete control over how data is collected, and so they can build these systems to build it to collect it consistently, to make sure that it's clean from the very source, and to they basically have end-to-end control. In biology, biopharma, it's the bench scientists who are really collecting the data, and so as an engineer, you have to get used to this fact that you don't have that complete control, and it's as much about working with the biologist to make sure they're collecting things the way that you want them as it is about what you do with it once you get the data. Then if you kind of bring into that the fact that in most tech situations, data is kind of coming in, one data point at a time, streaming, or however, it's a user clicking on something or whatever that is. In biopharma or in any biological setting, typically data comes in batches. An experiment gets run, the plate comes out, you get the readout from it, and so that means that the way that you capture it is very different. There's often a lot of the time is in the biology side of things before you actually get that data. So it comes in sporadically. And there's, especially in early discovery and exploratory work, the way that the experiment is set up is completely different each time, which may mean, different columns in the Excel sheet, not just because people are naming things differently, but because, there's different parameters that they're following. And so you have to get used to this idea that you're working in small batches, small kind of one-off often batches. And of course, there are assays that get run regularly and that get productionized and automated. But that's only half the picture, and in some cases less than half the picture.
Yeah, that makes a lot of sense. So, you go from an environment where you can see what's happening, where like humans are in control over the data generation function and in control over how they measure it, to an environment where we're trying to understand the data generation function as we're trying to design the method of measuring it most effectively.
Yeah, that's right. You have both the biology is, that is kind of doing what it wants to, and also the bench scientists are maybe not doing only what they want to, but they're trading off what the requirements of the lab with the requirements that are coming from the downstream data engineers.
Yeah, that makes a lot of sense. So you've been writing about biofoundation models a lot recently, and there's a bunch of different flavors of biofoundation models. You've got protein language models, multimodal models, clinical data models. For listeners who haven't been exposed to the full breadth of biofoundation models, or maybe haven't wrapped their heads around the space, can you walk us through the main categories of foundation models that exist in biology right now, and what each type is designed to do?
Yeah, absolutely. And I think one thing that I find really exciting about foundation models is, so when I started, not that long ago, 8 or 10 years ago, in working, doing this stuff in biology, doing software engineering in biology, there was this idea of building kind of multi-scale models and systems where, you could do it at the molecular scale, and then at the cell scale, and at the, organism scale or organ scale, or, there's multiple gradations in there. And it felt very abstract and kind of like more about marketing than about actual, like I think the folks who were deep in the research had a conceptual idea of what this meant. But as an engineer who was trying to actually like, write the code in between, it was very clear that like the actual engineering wasn't there yet. Foundation models, I think, have actually, at least for me, they've given me a glimpse of like what this actually could look like, like a technical, implementable, usable form. And that's because the transformer models are this very general and also powerful infrastructure that can actually that actually allows you to build models that link these different scales. So the kind of the smallest scale is of course the molecular scale, and so this is models like AlphaFold or Bolts, or there's an OpenFold project, which is like an open source version of AlphaFold. And these are essentially trying to predict interactions between individual molecules. Now we think of AlphaFold as predicting protein structures because that's kind of where it started. But really the only purpose of predicting the structure of a protein is to understand what it binds to, whether that's small molecules or other, other peptides, things like that. So I think of AlphaFold, or that type of model, as really being about predicting, binding between different molecules. And in fact, there's been other results recently that kind of show that you can just go directly. You don't actually really need to predict the structures. You can actually just predict binding from these molecules, and actually be able to take a protein that you only know the sequence, you've never actually, you haven't found the crystal structure. You could actually predict which small molecules or other types of peptides that it would bind with just de novo, having never actually even synthesized it, let alone assayed it. So that's kind of the foundation, and that is clearly valuable if you can make these models accurate enough. That's very valuable on its own. But as we all know, or anyone who's worked in this space knows, predicting individual protein, small molecule binding on its own it's important, but it's of still limited use. And so the next layer above that is, how do you go from understanding these individual interactions to understanding a cellular response? So this is kind of, the starting with RNA-seq, so understanding RNA expression levels in a cell. If you introduce a perturbation, whether that is, knocking out a gene or knocking down a gene, or maybe a small molecule that interacts with multiple, DNA, RNA, protein, if you make one of those changes, what are the downstream effects? And so, classically, this would be a kind of like a Bayesian model where you mechanistically, you have your signaling pathways and you mechanistically model that. Turns out those signaling pathways aren't, that way of thinking about things just isn't accurate enough to consistently do this. And so what you can do instead is basically use a neural network, take all of your RNA sequencing data, look at a bunch of different perturbations, and you can actually predict, fairly accurately what these downstream effects will be without understanding the individual mechanisms. And more recently, these models have actually also are using the same kind of transformer technology as AlphaFold and these molecular models, which means that in theory, in at least in the academic literature and practice, you can actually link these two together, and you take a de novo protein, or a sequence, you predict something about it in like at the molecular level, and then take that directly into the what's often called the virtual cell model, which is at this interactions level. And then of course you can kind of keep going and say, okay, now what about a tissue that is that has multiple types of cells, or an organ with multiple tissues, and of course a patient, and then get to the population level. And so these foundation models allow us to kind of link these, at least in theory. In practice, we're not there yet, but there's definitely progress towards linking them all together. And so I think there's a future, which maybe this is 10 years off, maybe it's 50 years off, but like there's a path to getting to where you could take a de novo molecule or a de novo protein, something that's never even been synthesized, let alone assayed, and actually predict what would what would the population response be? If you have a diverse population with lots of different genetics, what would it actually look like? So this idea of virtual clinical trials where you can have, thousands of novel compounds and actually run a virtual clinical trial on each one, before it's even been synthesized. I mean, obviously very far down the road, very far off, but I mean, the potential impact is huge.
The sort of the way that you segment them in terms of like the levels at which the models are being used makes a lot of sense. I think the one of the questions that comes to mind for me is sort of like, how do you see biotech organizations navigating that landscape in order to select which model or combinations of models should they be using in order to accomplish their particular task?
Yeah, I mean, I think we're in the very early days. Obviously, this is, there are companies that have built these models and are trying to sell them. I think pharma adoption is still, very minimal. Which makes sense. This is untested technology, and of course, like the economics of running clinical trials means that there's no way to know for sure what's going to work and what's not going to work. So I think adoption of like the molecular level models, I think is starting to pick up definitely. And again, there's an immediate application for that, right? Like if you can run binding prediction, lab teams or biopharma knows how to use that, knows, you're basically doing virtual screening, which is something that's done in the lab now. It's very easy to just say, okay, we'll do a model that will narrow down the list of compounds or peptides that we're screening. When you get to virtual cell models, adoption has been, I think, much lighter, just because they're I think the potential of them, once if you can get virtual screening to work, where you can start predicting these interactions more accurately for novel targets and novel compounds and all that, then the virtual cell model becomes very valuable because it basically gives you a more holistic prediction from that. Right now, using a virtual cell model on a novel compound is essentially impossible because you need to assay it in order to know what it interacts with. And so they're typically used more for kind of secondary tasks like target identification. They're being used more in a little more in clinical things like biomarker identification and patient stratification, because you can kind of understand how the genetics interacts with a known compound. But I think it's going to be a little while before they start getting used more in early discovery, which is, to me at least, where I think the most interesting potential is. And then the kind of the higher level, patient models, like digital twins, those are getting used a lot more in the clinical space, where a lot about the compound, you've assayed it, maybe you've even run a first in human.
Yeah. No, it's interesting that sort of as the technology evolves, part of that diffusion process, as we're all using more AI and machine learning models in our work, the part of the diffusion process is figuring out like what the new process looks like with the advent of these foundation models, and sort of what capabilities get unlocked by reorienting your processes in different ways around these models.
Yes, absolutely. I think if this works out in if the direction that this is going in continues, I think drug discovery could be radically different, in the future. It's just a question of whether that's in 2 years or 5 years or 10 years or 20.
Yeah. So, one of the things you pointed out is that these models sort of change the value equation for data that most biotechs have historically treated as throwaway. So, can you unpack that idea? Like, what kind of data are we talking about, and why did it used to be rational to ignore it?
Yeah, so I think the kind of classically, the way that, at least in early discovery, the way the data was collected is you have a specific question that you want answered. You design an experiment to answer that one question, and you answer that question, then you move on. And until recently, there was typically no way to use that data for anything else, just because it was collected in a fairly narrow context, and right in like for that specific question you're trying to answer. At the same time, um, if you wanted to make that data reusable, you would have to store it in a very kind of strict format. You'd want to make sure that like the schema was the same across multiple essays, which is again very difficult if the protocol is changing between runs. You would want to take the time to make sure you name it in a way that someone later would be able to recognize it, which is actually like legitimately difficult to do if you don't know what the use is going to be for, right? It's not just about biologists being lazy. It's a hard problem. So both sides of that, value cost equation have basically switched, right? So now, on the one hand, the cost of organizing data is going way down just because we have these LLMs, these agents where you can point it at a directory and say, hey, just like find the data for me, right? And now it's not perfect. If the information isn't there or if it's ambiguous, then it's not going to be able to do that. But the bar for what makes it findable and organizable for an LLM based agent is much lower than what it would, otherwise it would take like a person weeks, months to go through all that data and organize it. So on the one hand, the cost is much lower. On the other hand, the goal of a foundation model, or one of what the reason they're called foundation models, is because you can basically train a core part of the model that can make multiple predictions. So if you have 5 different types of assays that you've only run a few times, you can basically train the model to predict those readouts, and train one core model that predicts all 5 of those. And so now that core model is being trained by 5 times as much information, roughly, as if you had trained 5 separate models. And so you see this with, we were talking about with the molecular interaction models, where they can predict structure, but they can also predict binding, right? And they can also predict other properties. And that's what makes it a foundation model is you have this core, set of weights, this core part of the model that is kind of used with some additional layers on top to make these other predictions. And so that means that data that previously you wouldn't have enough of it to build a reasonable model, now you can combine it with all your other assays, or at least in theory, you will be able to do that in the future, and actually get some value out of it. So the cost of making it accessible to that model is much lower, and the potential value of that you get from it is much higher.
Yeah. Do you have an example of a place where you've seen this done?
So I mean, certainly, so again, with like AlphaFold, where you have these multiple endpoints, I think that's that's kind of the canonical example of, again, it can predict structure, and so it was, I think, AlphaFold 2 or 3, I forget. And then the Boltz model kind of took this even farther, where you can actually, once you predict it on structure, then you put in some binding data, and now you can find that it can actually predict binding on novel, unseen proteins. So that's a separate endpoint, right? That's a different kind of assay that you would run. I think there's other like toxicity kinds of predictions and things like that as well. But yeah, that's the classical one. I know, like Eli Lilly's Toon Lab, they started out with individual models for each of these different endpoints, for lots of different kind of small molecule endpoints. And my understanding is that they're also kind of planning to build that that foundation model that will then be able to use all that data across all those different endpoints. To build one foundation model.
Yes, if I'm understanding correctly, there's kind of two ways. Two ways this can go. It's like you have assays that you've developed that are sort of labeled data that you can then fine tune these models that have already brought in a lot of information about about your particular biological domain into the model, and so like you can use transfer learning, use fine tuning to get better predictive accuracy, or in silico predictive accuracy in what you would have previously needed thousands or hundreds of thousands of assays to be run in order to train your own model. And then there's also the situation where like if you're able to use the data that you have as inputs to the model, then maybe the model backbone itself can sort of fill in the gaps of your understanding, that your assay can give the model enough signal that it will be able to tell you, for example, that this molecule is close to other molecules in molecular space, or that this predictive endpoint that has already been pre-trained is likely to, it's able to add signal that you wouldn't have been able to bring from the information that you have yourself.
Yeah, that's right. Yeah, I mean, to get a little more into the mechanics, essentially, what the foundation model is it learns how to assign, these high dimensional vectors to every, say, small molecule. If it's a small molecule model. So if you let's say you have three targets that you have screening data for, against thousands of compounds, you can, if you train that core model on those three different endpoints of the different targets, it basically will learn how to, like, where to place the individual molecules in this core model so that the similarities and differences that matter, the kind of features that matter, are, well defined, and the features that don't matter are, basically ignored. And so it'll place them in a way where the kind of geometry of those embeddings is meaningful to how the molecules function. And so now if you have a third target and maybe do a smaller set of screening to kind of learn a little bit about that, well, now because it knows about the relationships between the small molecules, it can use that to predict, what unseen molecules will do in this new target. Um,
Yeah, that makes a lot of sense. So, to zoom out a little bit, you draw a distinction between sort of two types of biotech companies. There's ones that like are built around a single core assay that they're productionalizing the data pipeline for, and they're running over and over and over again. And then there's the ones that are more science driven, and they're running sort of whatever assays the research demands. Can you sort of outline how the foundation model opportunity looks different for those two different types of organizations?
Yeah, so certainly the kind of the obvious one. So yeah, and I should also add that those two types of organizations, sometimes it's about time. Like sometimes it's, like early stage startups, they're just building that first assay, and they kind of morph from a, very discovery focused, exploratory company to a more of a, core assay company. I mean, even those core assay companies, just because they have one or, or two productionized assays, doesn't mean they don't have that long tail of smaller assays that they're still running as, secondary exploratory assays. Yeah, so for those, if you have a core assay that you're running and you're building, terabytes of data from, then obviously it's relatively easy to, relatively to build a model around that, because you have that core set of data, tons of it. For a smaller company where you're or a company that doesn't have that core assay, where you're doing more of this exploratory work, really the question is, like, is there a way to basically get more out of that investment, because it can still cost millions of dollars to run all those smaller assays. And so the foundation model basically allows those companies to, or it gives them the opportunity, and maybe I expect this will become more so in the future than it maybe is today, but basically to take all those assays that they've run in this exploratory setting and, turn that kind of bootstrap, a foundation model or some kind of model, that will be able to use that, right? For the core assay companies, there's always this question of, do they have enough secondary data to like be more than a drop in the bucket in that core assay, but yeah, so I think it's a big opportunity for the companies that don't necessarily have a massive consistent data set from a core assay.
Yeah, that makes a lot of sense, and also it sort of gives more runway for people to like explore the space around the assays that they've already developed. So, I've in your Substack, you've made the argument that the answer for these labs is not more engineering; it's more SOPs, standard operating procedures. So, regardless of which type companies fall into, you've argued that the reason that data falls apart at a biotech organization at science-driven labs isn't technical. It's that biologists and data scientists fundamentally disagree about whether structured data capture is worth the effort. When you go into a lab and build a data operations plan, how do you bridge that gap?
Yeah, so I think sometimes I think it's hard to distinguish between when they're disagreeing and when they just have a different understanding of the situation. Like, I think it's very easy, especially for engineers who are coming from traditional tech or who haven't, had as much experience working in a biotech lab, to overestimate how much it's actually possible to automate and standardize these things. And so certainly I think there's some disagreement there. I think on the wet lab side, I think it's often more about not understanding, like, what's um sort of what's possible, or like expecting that it's going to be too difficult to do these things, or just not knowing what the expectations are, right? Because especially if you come out of an academic lab, it's the expectations for what to do is low, and so it may not be that they don't want to do those things or they're unwilling to. It's more they just don't know what's expected of them. And so I think on the one hand, there's this kind of cultural dynamic where the digital folks, the computational biologists, data scientists, data engineers, they don't feel like they can ask much of the biologists because they know that they're busy, and maybe they've had some experiences that weren't so positive in the past, and so I think it's very hard for them to ask those things. They also maybe don't understand the way the lab works well enough to really even know what they would ask for. And then the bench scientists, they're not being told what the expectations are, and so of course they're just going to do things the way that they've always done them. Sometimes you may not even notice. I was I certainly know bench scientists who think of like the ELN, the electronic lab notebook, as like the golden record, and so they like write everything down in a doc somewhere, and then only when it's perfect do they copy and paste it into the ELN. Whereas like the computational biologists, the data engineers, they would say like that's a terrible idea. Like it should be in there from the beginning so you don't forget it, right? But they may not even realize that this is happening. They just know that there's some breakdown somewhere. So I think it's often I think there is actually a lot more common ground that people often realize. So it's just a matter of kind of being more deliberate about saying like, these are the expectations. This is why we need you to do it that way. And I think in many cases, you can actually make it easier for the bench scientists to, especially now with LLMs and whatnot, you can make it actually so it's less work and less cognitive load. It's less, decision fatigue for biologists just to kind of follow the instructions if you give it to them. But a lot of organizations, are set up in a way where that just doesn't happen.
Yeah, so you call what you deliver a data operations plan. Can you walk us through what that contains? And if I'm like a scientist in a lab that just signed up for one of these, like what changes about day-to-day workflows?
Yeah. So, I mean, at its core, like the kind of the working part of the data operations plan is essentially just a collection of docs with instructions of when this type of readout, comes off of the machine. This is what you name the folder. This is what files go there. This is what information from the ELN we want it, or even if it's just like an ID. This is how we connect it to the ELN entry, whatever that is. It's just very simple instructions, and behind that's built off of fairly careful analysis of what kind of information needs to be collected, what are the assays, what's the right naming scheme, and so it's a fair amount of upfront work to gather requirements, put together a design, collect all of that configuration, but that upfront work, the goal is to make those instructions as simple as possible. And then if you create a reasonable list of, a reasonable SOP, a process for collecting that data, then it can actually be easier for the bench scientists to follow that, because they don't have to think about, okay, what do I name this? What folder should I put in? How am I going to remember that it's here? But yeah, but that's really all it is. And as an engineer, I often feel kind of sheepish that like this is the solution. It's it's not in running in AWS. It's in Notion or whatever Microsoft Word. In practice, especially for this long tail of assays that they're not run often enough to productionize. They're changing every time. Like you just can't write code to handle that. You have to address the human side of things.
Right. And it strikes me that, given we were just talking about biofoundation models, like there's an element of data strategy here too, right? Where you're trying to like understand sort of how to navigate all of the different trade-offs between schema detail and ambiguity, between the level of burden you're placing on the bench scientist versus your bench scientist's ability to execute on that, things like that. Am I thinking about that right?
Yeah, absolutely. And really, like, I think sometimes like it's very easy to over engineer these things because, again, with like as LLMs get better at, finding data, searching through, and so as LLMs get better at that, it's very easy to sort of think that you actually need to be more consistent, more precise than you actually do, right? So I think it's important to make sure that the information gets there and that it's unambiguous, to an LLM or to a person using an LLM to help organize it. But if like a few years ago, having a column name where sometimes it's capitalized and sometimes it's lowercase was just like horrific, right? Like it would be a huge pain to go through and fix that, to write that into scripts. Nowadays, like LLMs can figure that out, right? If you have a column name that could mean two different things, right? That's still a problem, but that's like a problem even if you had months to go through all the data carefully, that would still be a headache.
Yeah. So, I mean, just to think through this a little bit, it's not about, you knowing in advance exactly what question you need to answer using the data, or even, how that data specifically needs to be structured. It's about sort of like maximizing the information content of the data that you're collecting, and then like thinking about how you might want to expose it to agents or to the LLM so that it's accessible at the point when you ask the questions you don't know you're going to ask yet. Am I thinking about that right?
Yes, exactly. And moreover, knowing that like the LLM technology is probably going to continue improving, at least like slowly. Like you could argue that we've kind of hit a plateau there, but it is still improving. And so we kind of, you want to design both for what's realistic today, but also what is likely to be realistic, in a year or two.
Yeah. And like, as you've been doing these data operations plans, or you're going into organizations that already have sort of standard operating procedures or really strict guidelines around, what data needs to be collected and how it gets collected, are there any common failure modes that you've seen, or anything that you've learned from sort of watching organizations that do this in a bad way?
Yeah, so I think there's definitely like there's a spectrum here from like over engineered to under engineered, and you see folks on both ends of the spectrum. Sometimes you see individuals in the same organization on both ends of the spectrum. So I'm definitely have talked to worked with plenty of data engineers who have discovered the hard way that schemas are brittle, right? And if you say like here's the set of columns that every single assay needs to include, then the next week you find out, oh, someone wanted to run it with two compounds in the assay instead of just one, or there's an extra time point or whatever it is. Like whatever assumptions you make about how the assay is going to run will immediately be broken, and that will break your schema. So over engineering is definitely a problem. And then on the other end, under engineering side, I've definitely cases of where data went missing for months. There was a large scale screen. The team wanted to go back and like reevaluate it, and they suddenly realized that no one actually knew where it was. Probably the people who had run it had left. In one case, we had an engineer, a Russian-based engineer contractor who had worked with a company when they ran the assay, stopped working with the company. We happened to like start working with them again, and they were the only person who knew where the data was. So they found the assay because the team happened to start working with them again. So yeah, so that's obviously like an extreme case, but it does happen that this data goes missing. Every organization I've talked to has had at least one case where they had to rerun an assay just because they couldn't find the original data.
Yeah. And it like, on the other side of the coin, if a lab gets this right, like they put a data operations plan in place today, how does that enable them over the long haul, uh, as they sort of grow and mature as an organization?
Yeah. So on the one hand, I mean, I would argue that it has immediate benefits in that the bench scientists don't have to think about this, like, right? Like, and it's not necessarily even about the time, right? It's really about like that, the kind of the cognitive load. It's about, decision fatigue. It's about allowing them to think about the scientific side of the data and not about like what folder to put it in. And even if it's small, it does, make a difference over the long period. Then I think down the road, when you actually need to find and use that data again, it's going to save you not just time, but also, cognitive load. It's going to save a bunch of conversations of people going around and like having to ask like, hey, do you know where this data is, right? And so it can even if it's just a few minute conversation here and there, it can take weeks to like track down all the people you need. So it can massively reduce timelines. And then I think as this is kind of more in the future, but I think as biofoundation models become both more powerful and more flexible for the kinds of data that they use, I think that data that these companies are collecting will become more of an asset that allows them to, build new capabilities, that like we're probably not there today, but if you're a startup founder and you want to convince your VC partners that that you're actually building a moat, right? Like data is the moat, and this is how you make sure that you can actually leverage that in the future.
Yeah, no, this is something that I know you've written about before, previously as well. So I'm just interested in your perspective on it. Like you said, data is the moat. How do you think that like the quote unquote moat in biotech has evolved? Like with the emergence of these foundation models and with sort of like the shift in the biotech space to where there's a lot of more pharma driven partnerships, stuff like that.
Yeah, it's a great question, and I think it's constantly, evolving, right? You have companies like I think it's Tahoe Therapeutics that where they're basically all they're essentially doing is building up this massive data set of single cell data, and that's basically they're from what I understand, essentially want to get acquired by a pharma company just for the data set, right? So they're very literally just building a shell around a large data set, and that is their moat. And then you have companies like Eli Lilly, Toon Labs, where they've basically they're building a bunch of models around $1 billion worth of data that they've collected, and they're uh, more or less giving it away, right? It's kind of a partnership where you have to give some data back, do federated learning, all that. But so for them, they're kind of seeing the data as currency to basically build relationships with companies, with the startups that they may want to invest in or acquire someday. And so and in between you have a lot of different models for this. I think it's still playing out, right? And what we're seeing is that it's not just about the data, right? And it's not just about the models, because models are just code. It's all open source these days, right? Like the data, like the model without the data is kind of useless. But if you can find the right way to merge the two, where you can build the model that makes that is able to make predictions with the data that you need, if you can find that right combination, then like that's where the value is.
Yeah. No, it's an interesting time, but regardless of where it evolves, like thinking deeply about the data that you're collecting and about the different sort of the optionality that data gives you as the ecosystem evolves for how you can use that data depending on the type of company that you're becoming and what your revenue model looks like. I think it seems like a really good idea to think deeply about that up front.
Yes, absolutely. Well, and also to recognize that like those options are like changing, and there's more options for how you leverage that data every day. And so yeah, I think it's useful to kind of hedge your bets by making sure that that data is accessible and unambiguously organized, so that whatever that option, whatever options open in the future, you can take advantage of them.
Yeah. Last question before I let you go. Obviously, sort of the emergence of biofoundation models, and also, companies focusing very deeply about, like on the data that they're collecting, and the availability of LLMs and agentic AI sort of it changes the dynamics of the software ecosystem for in the biotech space pretty deeply. So I'm just interested in, you know, what are your opinions on ELNs in particular, and sort of like how the software ecosystem surrounding your growing biotech organization is going to look?
Yeah, I mean, there's a lot of people who have kind of prophesized that ELNs are, their days are numbered, that they're not going to be useful anymore. I think they're definitely like if you're in an automated lab where robots are doing everything, then yeah, you don't need an ELN if everything is automated and basically it's all metadata. But I don't think, at least I don't see a future in which we completely move away from scientists with pipettes in labs, just because there's always going to be a lot of exploratory work. There's always going to be these assays, which again, is you don't run often enough to automate them. And I mean, maybe at some point the automation gets capable enough that it can run any assay one off, but I don't think we're anywhere close to there now. And as long as there are humans in labs with pipettes, like the ELN is the tool that they need. And so I guess Yeah. So as long as there are humans with pipettes in labs, I believe ELNs will be a necessary part of that.
Yeah. No, I mean it's interesting, and sort of like to the analog for for industries at large is, as long as there are humans picking up phones to dial customers, then there will be a CRM, regardless of whether of whether agentic AI can take care of everything. So yeah, it's interesting to think about sort of which of these software systems are sticky and why they're sticky based on where we think the value of these systems actually is.
Right. Yeah. Now there's another question of whether every lab will just build their own ELN from scratch using Claude Code or Cursor or whatever, which I also don't think will happen, but that is a much longer discussion.
Yeah. Well, and I've seen like some labs that I respect sort of saying, we're just using Notion now, so like and sort of like the extreme flexibility of, a back office software platform that's meant to like serve as your documentation record, and, being more comfortable with everything being a little bit more unstructured versus, having an understanding of your workflow. It's going to be interesting to watch people sort of continue to navigate that like schema rigidity versus ambiguous and more like free flowing.
Yeah, absolutely. Yeah. So maybe that's more likely to happen than ELNs going away completely. But I think it'll be interesting to watch that space.
Totally. Well, Jesse, thank you so much for coming today. I really appreciate you joining today.
Yeah, thanks so much for having me. This was fun.
Jesse, thanks for coming back on. I want to pull on a couple of threads from this conversation. The first thread is that your data strategy has to account for the fact that the value of your data is going to keep changing in directions you can't predict. As we discussed, the data you collect to answer one specific question gets repositioned by foundation models to answer questions you never thought to ask. Tahoe Therapeutics, an example Jesse mentioned, is building an entire company that's essentially a shell around a single cell data set, betting that the data is the core asset. As we discussed in a previous episode, Lilly spent $1 billion generating screening data and is giving away access to models trained on it partially as currency to build relationships with startups they might acquire. Two completely different bets on how data creates value, and both only make sense if you believe the utility of that data is going to compound in ways that nobody can fully map today. And the second thread is the distinction between consistency and ambiguity, which sounds small but changes how you plan. LLMs have made inconsistency a non-issue. Things like mixed capitalization, different file formats, CSVs and JSONs in the same folder, that doesn't matter anymore. What still kills you is ambiguity. A column name that could mean two different things, a file that you can't tie back to an experiment, or tacit knowledge that is loosely documented or not documented at all. That's the line your data operations plan needs to hold. And based on what Jesse is proposing, it's much more achievable than many realize. You can find Jesse on LinkedIn and on his Substack, Scaling Biotech, where he writes about this every week. If you know someone at a biotech sitting on scattered assay data, wondering whether it's worth the effort, send them this episode. And please subscribe to Data in Biotech wherever you listen, and we'll see you next time.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.